Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture we are going to talk about the nucleic acid or the central dogma. Okay, so let me write nucleic acid or central dogma. Central dogma. Okay. This central dogma and nucleic acid are very important because in the previous video lecture, if you guys remember, we talked that DNA is actually a genetic material, or we talked also that RNA is actually the genetic material. But we didn't talk that how DNA will be look like, how RNA will be look like. We didn't talk the structure of the DNA. We didn't talk the structure of the RNA. So for the structure of DNA and RNA, we need to understand that the nucleic acid, we need to understand the central dogma. Usually what actually happening inside our body, every cells have their central of dogma. Now what does it mean? It means that we have the DNA will be look like in this form. We will be explain the detailed structure of the DNA inshallah in our upcoming video lecture. This is the DNA structure. Will be look like a double helical in shape. Okay, usually DNA is also a single stranded, but usually Watson and Crick describe that DNA is a double stranded, and inside the virus DNA is also double stranded and single stranded, but we will talk it. We already talked that DNA is also single stranded. Okay, you can watch it. Now then, from the DNA, what's actually going on. From the DNA we get the RNA, and we call that mRNA. Okay, messenger RNA. We call that phenomena transcription. We'll be talking later in a detail. Then from the this messenger RNA we get the proteins. These are actually a long polypeptide chain. We get a protein structure. So we call that translation phenomena. Okay, sometime what actually happened, we also get the exact DNA copy as well. From that DNA, which we call that parental DNA or old DNA, we also get another DNA which we call that daughter DNA or new DNA strand. And whenever we get the DNA from the same DNA, we call that replication. Okay, so this whole process is called as central dogma of the life. We will explain the replication phenomena in a detail. Replication phenomena is also happening inside the prokaryotic cell and inside the eukaryotic cell, but it's different in prokaryotic and in eukaryotic. We'll be seeing in a detail in our next video's lecture, in our upcoming video lecture. Then we will see about the transcription, that transcription is also different inside the prokaryotic cell and inside the eukaryotic cell. Okay, we will also see inside the prokaryotic cell the transcription inside the eukaryotic cell. Similarly, we will see the translation phenomena, okay, inside the prokaryotic cell and inside the eukaryotic cell. All these things are actually known as central dogma of the life of the cell as well. Okay, so this is very important for you. Now the second thing is that what is actually nucleic acid. We need to understand these things, but before going to explain the whole central dogma, we need to understand that what are actually nucleic acid, because for this understanding you need to understand the basics, and the basic means you need to understand the nucleic acid. Nucleic acid, if we see, so nucleic acid, let me write in a short form. Nucleic acid are actually the polymer of DNA as well as the RNA. DNA and RNA polymer. Okay. DNA RNA polymer means that there's a long chain of DNA as well as there's a long chain of RNA. Both are usually present inside the nucleus. From DNA we get the RNA, and then RNA is translated outside the nucleus. So both are actually present inside the cell, but not RNA is always present. From DNA we get the RNA. Okay, we will be talking inshallah later. So both are present inside the nucleus. That's why we call it nucleic acid. And why we call it polymer? Because both are the long chain of the nucleotides. Okay, these are the long chain of the polymer which we call it nucleotides. Now what are the nucleotides? Nucleotides contain pentose sugar, phosphoric acid, and nitrogenous bases. Okay, so let me write. Let's try to understand these three things, that what are actually these three things, and these three things actually make the nucleotide structure. These are actually the basics. You need to understand these basics, means the nucleic acid basics. Once you get the idea about that, then the central dogma will be very easy for us. The understanding of the central dogma very easy for us. Let's try to understand. Let's first see that what are actually nitrogenous bases. Usually the nitrogenous bases are the adenine, guanine, cytosine, uracil, and thymine. These are called as nitrogenous bases. Usually, if we see, so nitrogenous bases are classified into two forms. One we call that purine, and the other we call that pyrimidine. Purine and pyrimidine. Inside the purine we have the nitrogenous bases. You know, purine are actually a double ring structure. Purine are the double ring structure, means they will be look like in this form. Okay, so double ring. And pyrimidine are the single ring structure. In purine we have adenine and guanine. Adenine and guanine. Okay, and inside the purine we have nitrogen atom as well, but we have almost 4 nitrogen atom in case of purine. Okay, and the position of each nitrogen atom will be placed on carbon number 1, carbon number 3, carbon number 7, and carbon number 9. Okay, so we have 4 nitrogen in purine. You know, one thing that I forget to mention, purine are the double ring structure, and we call that dicyclic. Okay, then we have pyrimidine. In case of pyrimidine, we have the nitrogenous bases, we call that cytosine, uracil, and thymine. Sorry, they have 2 nitrogen atom. They have 2 nitrogen atom which are placed at position number which are placed at carbon number 1, which are placed at carbon number 1 and carbon number 3. Okay, and as it is contain single ring structure, so we call that monocyclic. Okay, so these are actually the nitrogenous bases. Usually in this case, the adenine, which is the nitrogenous bases, they make a bond with thymine, and as well as they make a bond with uracil. Okay, adenine will make a bond with thymine and with uracil also, and they make double hydrogen bond. Okay, 2 means double hydrogen bond. They make a hydrogen bond with thymine as well as with uracil. And if we see cytosine, so they make a bond with thymine. Sorry, cytosine will make a bond with guanine, and they usually make triple hydrogen bond with guanine. Fine. So these are the nitrogenous bases. Now let's see the structure of the phosphoric acid, that how exactly phosphoric acid will be look like. So 3 hydroxyl group we have. Okay, now the phosphoric are always belong to in periodic table it is belong to fifth number, so that's why they have five bond. So 1 2 3 4 5. Okay, fine. This is the phosphoric acid. Okay, these are the phosphoric acid. Then we have pentose sugar. Okay, now what does pentose sugar means? Pentose, as the name indicates, pentose means, penta means 5. O's can represent the, if we see the spelling, full spelling of the pentose. So pentose sugar. Here penta means 5. O's can represent the sugar properties. Okay, now how exactly pentose will be look like? Pentose will be look like that they contain 5 carbon ring because it is called pentose. Okay, now here is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and here is the carbon number 5. So that's why we call it pentose sugar. And they have OH, sorry. OH, and here is CH2OH. So this is the complete structure of the pentose sugar. Fine. Inside the DNA we have this pentose sugar. Inside the RNA we have also the pentose sugar, but different. We'll be talking later. Now this is the pentose sugar. Now whenever we combine all these things, means when we combine the nitrogenous bases, when we combine the phosphoric acid, as well as when we combine the pentose sugar with each other, then they make the nucleotide structure. Okay, so that's all about the nucleotide. Now how exactly the nucleotide will be look like? Although I draw each of the structures separately, but combinedly how it will be look like. Now let me draw. This is the pentose sugar. I hope you get the idea about the pentose sugar. Usually at carbon number 1 we have the nitrogenous bases. Fine. At this position we have what? We have the nitrogenous bases. Right? It may be adenine, it may be guanine, it may be cytosine, it may be uricil. Anything we call it nitrogenous. So nitrogenous base is always present with carbon number 1, and they make a bond with carbon number 1. We call that glycosidic bond. Glycosidic bond. Okay. Now second thing is that they contain phosphoric acid. So phosphoric acid will be present at that position. Okay, so P, OH, and double bond O. But remember, OH, this one OH and this one OH, both are combined together and removed in the form of water. And they make a bond which we called as phosphodiester bond. Phosphodiester bond. Okay, now this is how the nucleotide will be look like. This is the whole structure of the nucleotide. So we have nitrogenous bases which is connected with carbon number 1, and we have phosphoric acid which is connected with carbon number 5. Okay, and carbon number 3 to these two things are free. Now as we talked that DNA has its own pentose sugar and RNA has its own pentose sugar. Now remember, when we talked about DNA, so DNA means DNA have ribose sugar, we call that deoxyribose sugar. But here at carbon number 2 the oxygen is removed, that's why we call that deoxyribose sugar. Deoxy means removal of oxygen. Deoxide means the oxygen is removed at carbon number 2. That's why this is now called as deoxyribose sugar. This whole structure is the deoxyribose sugar. Now whenever they have again the same thing, then we call that ribose sugar. So actually the OH is the functional group. These are what? These are the functional group. In this case, at carbon number 2, in case of deoxyribose sugar, they have, they have 1 2 3 1 2 3, they have 3 functional group. At carbon number 2, the functional group is not present. Instead of functional group, they have the hydrogen bond in case of deoxyribose sugar. In case of ribose sugar, we have 4 functional group: OH, 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 OH. Fine. Now that's why we can say that this ribose sugar less stable. So ribose sugar is always less stable. Fine. But DNA is more stable. Why? Because DNA have only one hydrogen. They have only 3 functional group, and at carbon number 2 they have 1 hydrogen atom. So we call that they are the more stable. They are usually more stable. Now second thing is that as they have functional group, means ribose sugar, so they are more reactive as well. More reactive. Okay, as they don't have the functional group at carbon number 2, so they are less reactive. Less reactive. Okay, now that's a little bit property of the ribose sugar as well as the deoxyribose sugar. Okay, in RNA we have the ribose sugar are connected to each other, means these nucleotides are connected to each other and they make the RNA. And in DNA we have the deoxyribose sugar are connected to each other and they make the nucleotide, means a complete nucleotide, and we call that DNA. That's the difference. So remember, whenever we talked about the DNA, so they have the deoxyribose sugar. Either the strand is single or the strand is double. That's very important things. We always heard that DNA is a double stranded. DNA is also a single stranded. It doesn't mean that we identify the DNA with the strand. Whenever we see the 2 strand, we call it DNA. Now, when you see the deoxyribose sugar, you will call that this is actually DNA. Either it is a single stranded or it is a double stranded. You must watch this deoxyribose sugar. We have RNA also. You may also heard that RNA, when you see the single stranded, we call that this is RNA. Now, it doesn't mean that RNA is always single stranded. RNA may have also double stranded. We already talked it in the virus genome. Okay, so when you see the ribose sugar, then you will call that RNA. If it is double stranded or single stranded. That's very important property. You need to understand about that. Then another important thing that we also heard this term as well. Nucleosides. We also heard this term. Nucleoside. Now what is nucleosides? Remember, when we remove the phosphate group with the nucleotide, we call that nucleoside. How exactly? For example, we have the nitrogenous bases plus we have phosphoric acid. We have a pentose sugar, and when we remove the phosphoric acid, we call that nucleoside. So let me once again tell you: nitrogenous base plus pentose sugar minus phosphoric acid, we call that nucleoside. The same structure we have, but when we remove the phosphoric acid here, we call that nucleoside. Okay, adenine have also their own structure. Guanine have also their own structure. You also word these terms also, means let me write here. You also word these things. ATP. You also listen this word ADP, GTP. A lot of the terms we have. Okay, ATP, GTP, ADP, TTP. These terms you may also see it in the book or anywhere, or you may have probably heard these words. What does it mean actually? It means that as we already talked, the adenine have a double ring structure, guanine have also double ring structure. Now remember, whenever the adenine have one phosphate group, when they have one phosphoric acid, then we call that adenine monophosphate, AMP. Okay, when they have two phosphate group, we call that adenine diphosphate. When this adenine have three phosphate group, we call that adenine triphosphate, and so on. Similarly, when guanine, this is also a structure of the guanine, but a little bit different to each other, we'll be talking later. Guanine have one phosphoric acid, we call that guanine monophosphate. When they have two phosphoric acid, we call it guanine diphosphate. When they have three phosphoric acid, we call it guanine triphosphate, and so on. Similarly, cytosine monophosphate when they have one phosphate group, cytosine diphosphate when they have two phosphate group, so cytosine triphosphate when they have three. Similarly, uricil, UTP, uricil triphosphate, mean this thing. And thymine monophosphate, thymine triphosphate, thymine diphosphate. All these things are actually their own structure. Okay, they are very complicated things. Not a complicated, but very easy. But when you saw the structure of these things, so it's looking very complicated. But here I'm not drawing the exact structure of the adenine, guanine, cytosine, thymine, uracil, but these are the structure of the nucleotides. Okay, these are the structure of the nitrogenous bases. You need to understand this structure also. Now, once you get idea about all the basics, now these are actually the basics. Once you get idea about the basics of the nucleic acid, and once you get idea about the central dogma, here is the central dogma, we also discussed a little bit, means the basics of the central dogma. Once you get idea about these things, then the structure of the DNA, replication of DNA, transcription, translation, everything will be easy for you. So these things are very important. These are actually the key molecules. These are the basics. You need to understand all these things. Okay, that's are very important. So similarly, we have also, if this is the deoxyribose sugar, we have also, if there is one phosphate group, we call it deoxymonophosphate sugar. Deoxymonophosphate sugar. If it is two phosphate group, we call it deoxy diphosphate sugar. As they have also nitrogenous base. Let's suppose if they have adenine, so and they have adenine and they have one phosphate group, and this is the deoxyribose sugar, we call that deoxyadenine monophosphate, because one phosphate group. If this is the two phosphate group, we call it deoxyadenine diphosphate. If there is guanine nitrogenous base is present, then we call that deoxyguanine monophosphate. So these are very important things. Inshallah, we will make a separate video lecture on all the nomenclature of these things in some other videos lecture. But you need to understand all these things. These are the basics. Okay, I hope you get idea about the basics of this nucleic acid, as well as you get the idea about the basics of the central dogma. Inshallah, in the next video lecture, we are going to see that how exactly the DNA will look like. So inside the DNA, we already talked that they contain the nitrogenous bases, pentose, sugar, and phosphoric acid, all these things. We will see that how the DNA, means how the nucleotides are connected to each other and they make a long polymer chain, which we call it DNA. We'll be seeing inshallah in the next video lecture. So I hope you get idea about this one video. So if you understand, so please like it, share it, and subscribe the channel, and stay tuned for the next video lecture, means DNA structure. Thank you so much.